microfabrication technology with the healthcare, and then give some general examples on the BioMEMS devices, and after that I will go to some particular example where you can do the cell characterization, biological cell characterization using the MEMS device, and that characterization is mostly the electrophysiological characterization. Okay. For the living cells, you can do for the physiological characterizations, and the biological aspect you have a various assay. You can go for the antibody antigen reaction to detect the biomolecules, how the cell growth takes place. It is now very standard techniques at in that domain, particularly in the biological aspect. Say this is the very standard technique, and there different assays are used to get the or to detect the biomolecules. Okay. Other than that, without labeling the molecules or the cells, can I characterize the living cell, okay, or you can I sense some particular biomolecules using the BioMEMS devices. Okay. So, that is my will be my lecture. The one project we have taken, and there you have to separate the cells, okay, the normal cell and the disease cells using some electrical technique, and then can I characterize those normal cells and the disease cells and see that what is the difference in the its properties. So, that is the main objective. So, we those things we will discuss okay, in the today's lecture. That will be an example for this what is the usefulness of the BioMEMS devices. So, first of all, it is a fusion of technologies in the micro scale. Okay. Photolithography, which is basically the heart of our microfabrication technique, and we have discussed in the last class. The medicine, it is in another domain. So, they were the total stranger to one another. At present, they are indispensable partners in the biomedicine to envisage the scenarios of the personalized medicine. Okay. So, now today's aspect, photolithography and medicine, that can be fused together to get the devices to sense the different biomedical signals, okay, and characterize them. So, what aspects the specific prevention and intervention strategies. Now, our society is going towards that direction. Various R&D are being done to get these specific prevention and intervention strategies. Early detection protocols to identify the disease when it is easily subdued. So, at the early stage, can I detect the diseases? That is the another challenge, particularly a very common example is a cancer. Okay. Can I detect the cancer at a very early stage, which is really very difficult still now. Okay. If we can do, then it lives can be saved. Okay. And the technology allowing for the long lives in the company of the disease as a good neighbor without sacrificing the quality of the life, that is the third aspect. So, that a long lives with the company of the diseases. Okay. The disease will be our a good neighbor. Okay. We should treat it in such a way, so that I can live a long life with a disease, but without sacrificing the quality of life. So, these are the 3 different challenges where now the medical domain and the technology domain, they are working on that too, and the technologies are being fused together to give this kind of comfortness. And here in this aspect, the development of miniaturized wearable implantable biomedical wireless sensors for personalized healthcare and the beyond hospital application. Okay. So, the personalized healthcare, okay, that aspect people are working. The another aspect is beyond the hospital application, because particularly in our Indian scenario, you know, the hospital are over crowded. Okay. And also in a remote villages we have a very few hospitals. How? And the patients has to be taken to the hospital, and then the disease can be diagnosed. But instead of that, can I do some technological innovation so that it can be a beyond hospital application, a blood test can be done where the in the home itself. Okay. That kind of things are being considered. Now, the another slide, it is on size and scale, why the BioMEMS is so much important. Okay. The MEMS has evolved because due to the advancement of the microelectronics technology. Okay. And therefore, we get the pressure sensor, temperature sensor, accelerometers. So, all those devices have come. Okay. At that time the the microelectronics technology was quite matured. People have utilized that and got the device, and the first commercialized MEMS was the pressure sensors, and it has the largest market globally. Second is the accelerometer. Okay. Those sensors or devices for the non biological or non healthcare applications. But why people are now getting interest in the MEMS or the for the healthcare or biomedical application of the MEMS devices? Because there is a similarity in the sizes, and the organization of the common structure between the micro nanoscale devices and the biological species makes this technology an obvious choice for getting the different devices for the characterization or detection of the biomedical signals. If you say here the MEMS device, it is this aspect, it is a top down approach. Okay. You take the substrate and go for the do the implement the device, and there you see the MEMS, it is the hundreds of microns to the 1 micron within this range. Then you have a MOSFET device which is in the submicron, and today and then you have a gate insulator, the thickness is in the nanometer dimension, that I have discussed in the last previous class. Coming to this other side, what are the bio species, the dimensions? Here you have a plant and animal cells within in that a few tens of microns. This is the RBC, the diameter of the RBC about 10 to 20 micron, not 10 to 20, it is the 8 to 10 micron, this diameter of the RBC. Bacteria will go mostly from the 1 micron to the submicron, then you have a virus going to the nanometer dimension, proteins, DNA, it goes to the nanometer dimension, and which meets towards the atom, okay, only the few angstrom. Now, if you see both the sides, you have the here the devices which will sense, and these are the different dimensions for the different biological species. Okay. The one aspect here that it is a top down approach by which we will get the BioMEMS devices, whereas if you consider these things, this is all about the naturally occurring materials or the living elements, where it is due to God's gift, it is a bottom up approach. Atom by atom, molecule by molecule, it grows up, and we get this biological species. Therefore, when we talk about the sensing of this the biomedical signals, we have to say at the cellular level or at the molecular level how we will sense this signal. For that we must have the device which for which the characteristics length should match, then only you can capture those signals. Okay. I told that you have a probe, just consider the lithography. You have a optical lithography where the wavelength of the light is much smaller than your feature dimension. Now, in the next, if I go down and going to the submicron to the nanometer scale, there this the wavelength will not be, cannot be useful, because you have a diffraction effect. So, there I have to reduce the wavelength, and therefore, it was completely switched over from the optical domain to the electron beam lithography domain, okay, because you have a very narrow wavelength which can be useful to get the nanometer dimension of the devices. So, the probe by which you are getting the features, or the probe by which you can detect the signals, the atomic force microscopy there also. So, that should match together, then only I can get the sensing signals properly, otherwise it will just be a noise. And that is the one important aspect for which this BioMEMS devices are becoming more and more important to us. You can get a very sensitive signal using the BioMEMS devices. You can use a very large device, conventional large device I can use it, but there getting the signal at the molecular level is very tough. It will simply be a noise. Okay. So, this is the another aspect which we should consider, and that actually leads to the development of the different devices. Okay. These are the some general slides. Now, the MEMS use the micro size component to develop the sensors, transducers, actuators and electronic devices to sense or to make something happen. Many of the MEMS used in the consumer product and the other areas are also found in the medical devices. Now, the BioMEMS means it is the MEMS device which will handle the biological species. It is as simple as that. Okay. But it is the MEM when it started its journey and developed the different devices for the application in the aerospace, can be in the agriculture, it can be in the environmental, have been found useful in the medical devices also. Just as a example, for the aerospace, you know that accelerometer, which is used to get the acceleration of the moving system. Can I use it for the medical as a medical device? It can be useful, and today it is being implemented in the different systems, okay, for the healthcare monitoring. Just think of for a elder very aged person, okay, if sometimes he may fall down, okay, a sudden fall down, can I detect it? Is if I have accelerometer sensors attached to his body, then it will detect that sudden fall and give the signal. Okay. Or how this heart is pumping, that we can monitor just putting a accelerometer over there. Okay. But obviously, the design aspect principally it will work in a similar fashion, but as I told the specification, in this case the range should be the different compared to the range that we will design for the aerospace application. Okay. So, that is the one aspect, several devices you can have, say for the environmental. Here different gas sensors are now available, CO2 sensors, oxygen sensors, or different other different gas sensors are now available. How the gas sensor works? Any idea? For the automobile also you have the gas sensors, how much carbon monoxide is being ejected, that can be calculated, and based on that you can have a feedback mechanism, so that the fuel can be controlled into the engine. Okay. But basically how the gas sensors works? Using a nanomaterial. Several gases are there, okay, say oxygen or carbon dioxide sensors. Basically you can have some material, which kind material, which will absorb, any material will absorb the gases, but I have to choose some material which is specific to this gas. Okay. I cannot detect the several gases at a time using a single device. I will have a device which will detect only one or two specific gases. Okay. Depending on the application, I will choose the material. Now, earlier you have the material means it can be a thin film form, it can be a bulk form, but today we are using the nanomaterial. That is for the zinc oxide. Zinc oxide nanomaterial can be useful. Okay. It can absorb the gases, but what is the importance of using the nanomaterial instead of a thin film of a zinc oxide or bulk silicon zinc oxide material, that will also absorb the gas. The yes, the important characteristics of the nanomaterial is that it has a larger surface to volume ratio. Surface area is much higher compared to the volume, and so as it has a more surface area, so the absorption of the gases will be much more, interaction of the gas with the gas molecules with the material will be much higher, much higher. So, the gases will be trapped in the material, and accordingly the characteristics of the material will changes, and that you will detect. In the electrical domain, we will just get the change in the electrical resistance, okay, that of that particular material after absorption of the gas molecules. So, by getting that delta R value, you can say how much is the what gases are present. Okay. That is the very basic principles. Now, you can modify your material, so that your range, sensitivity, all these things will changes, modifies. Okay. That is the basic concept. So, here also that gas sensors, can I use it in the medical devices? Yes, it can be useful, because for the medical purpose, for healthcare monitoring also we require the gas sensors. Okay. And there this kind of sensors are used. Okay. MEMS pressure sensors are found in the blood pressure monitoring, infusion pump, catheters, and several other applications also. Okay. So, it is a biosensors is a analytical devices that combine a biologically sensitive element with a physical or chemical transducer to selectively and quantitatively detect the presence of specific compound in a given external environment. This is the somewhat you can say it is a definition of the biosensors. Okay. Coming to the some general examples, here a micro cantilever transducer coated with the antibodies that capture a virus in the blood sample while ignoring the other component in the sample. So, here you have a array of sensors, okay, array of cantilever beam where the it is coated with the antibodies, okay. And then if the flow the blood through this cantilever, then it it will capture the virus, and by capturing the virus so it will the bends, and I can detect it by several mechanism, either using the optical method, or you can have a piezoresistor embedded at the at this end and get the change in the resistance value, or by the resonant frequency change. So, by several mechanism you can get what is the condition of this cantilever, and from that you can get whether the blood sample has this that virus or not. Okay. This is some ACM photograph showing the neuro probes. We sometimes we have to sense the electrical signal from the neurons, which is very thin, very small, as well as very soft also. So, as I told, these probes should have a dimension which is compatible to that neuron dimensions. Okay. Therefore, I must have some needle kind of thing where the tip dimension is much smaller than the in the submicron or the nanometer dimension. So, it can probe actually and get the biomedical signals from the neurons. Okay. And this is can be made, earlier it was fabricated using the silicon material at the initial phase, but silicon it has its Young modulus which is much higher than the neurons, and when you just go and touch that, it may damage the neurons. So, what way we can avoid this is that I can have this kind of a needle using a polymer material, where the Young modulus of the polymer is compatible with this Young modulus of the neurons, so that the damages can be avoided. Okay. The other side, it is the cell culture and analysis. The cell culture means it is in the biology. Here I have a audience, mixed audience. So, some of you are from the biological domain, some of you are from the engineering domains. Okay. How many are from the engineering? Many, okay, or at most half. And so, some of you know the biological biology and the cell culture, some of you know, but the engineering participants, do you know the cell culture? Some of you know. Here actually cells is a living element, then can I grow it? Yes, I can grow at a some controlled environment. Okay. And I can characterize the cell properties of when it is being the growth is being taking place. Okay. That is the, in general, that is the cell culture. And during the cell culture time, I have to analyze. Okay. The cells will give the signals, and I have to capture those signals and get the analysis. Okay. For that, generally the cell culture is done in a petri dish. It is a quite, that is a conventional method. Okay. It is a macro device. You put some media means it is a liquid where you have some nutrients, okay, and put some cells and keep it in some condition, ambient condition, 37 degree centigrade, carbon dioxide flows would be there, and as the days goes on, then the cell will proliferate, and while it is proliferating, then it will get the nutrients from the media, and then after some days it will proliferate on the whole petri dish, and after that it will decay, I mean death will take place and the cells will die. But during the proliferation and the growth phase, I can get the different assay, I can do the different assay for the cells for characterization, but that is mostly in the different assay what we do in the biological domain, put some antibody, antigen, see under microscope and get the results. while this proliferation of the growth is taking place, can I capture some non-biological signals from the cells, non-biological signals from the cells, so that those signals, it is the characteristics feature of the cells, and if I can get those signals, I can characterize, I can identify whether the cells is in a normal stage or it is in a disease cells. Okay. So, for that, instead of going for this petri dish kind of thing, which is a bulky, I can have a very small devices in a micro environment. There also cells will grow using the proper media and the ambient condition, and then there also I can characterize and capture the signals. Okay. So, for that different, these are the different cell culture devices. You can see where you have just a well, just like a petri dish, and you can put the cells over there and go for the cell culture. Okay. And mostly I will discuss this part in my subsequent slides. The other than that, it is a this is a one complete system which actually detect, it is a some company's insulin pump. Here you can see that the glucose will be detected. In the last class already I have discussed, and then depending on the glucose level, the pump will be activated and the insulin will be injected into the body. This is the whole system, okay, system in package, and now it is available in the USA and the Europe, but may not be available in the India, but it is available. Commercial product is available, and also I told that it is in a wristwatch form also it is available now. Okay. There are several other applications are there. It is difficult to cover all the aspects, this is just a few examples I have shown. Probably now I will go to my main that examples, as I told, I will go for that, where you will see that how the fabrication can be made, where I can do the cell culture, okay, and what are the biomedical signal that will capture. Mostly it is a bioimpedance. Impedance we know from the engineering aspect, engineering students you know the impedance. Okay. Basically it is the resistance to the current flow in the system. Okay. Now, the cell, we consider the living cell, and always in our mind it will have in the biological aspect, but if we consider it as a just a material, then it also has the impedance value, how the current will flow through that, and can I monitor the impedance during the cell culture. While the cell culture is going on, cell will divide and proliferate, and at that time I will pass the current through that, and I will monitor what is the impedance change occurring. And it has been observed that for the normal cell it will go some pattern, the disease cell it will have the different pattern, and from that biomedical, that bioimpedance signal, if you analyze, then you will get the cell properties, like what is the cell membrane capacitance, what is the cytoplasmic resistance, those properties also you can evaluate. Okay. So, here the hypothesis, main hypothesis is that sensing the non-biological parameters, proliferation of the cancer disease occurs due to the various physiological changes at the cellular level. Okay. So, I will start the case I have taken, it is a cancer disease. Okay. A normal cell, it will can slowly go towards the cancerous cell. Okay. And while it is going towards that direction, what is the changes occurring in the biomedical signals, and those signals, the bioimpedance particularly, it is we consider it is a bioimpedance, okay, in the electrical domain. So, at present the detection techniques are limited to the biochemical assay using the labeling of the disease cells. Okay. You can take the cells and you can label it with a fluorescent tag, and then if the cells will be tagged with the fluorescent molecules, and then if we observe under microscope you will see the fluorescent color. If those cells are not present, then you will not get the fluorescent color. So, that way these biochemical assays are being done, and that is why it is called the labeling technique. Okay. However, the mechanical, electrical, optical properties which we consider as a non-biological parameters of the cell also change during the various stages of the malignancy. Please consider this point. We have only considered the biochemical assay. Okay. The how it will be the fluorescent will be tagged with the molecules, because there is a antibody antigen reaction. Okay. The antibody antigen reaction will take place when the specific protein or the biomolecule will be expressed in the bioassay, biochemical assay, and therefore, those if that specific reaction takes place, specific molecules is expressed, then the fluorescent molecule will be tagged with that, and then you can under microscope when you will see you will get the light. Okay. So, it is all towards the biochemical assay, but while this is taking place, we can consider that the cell itself not only the biochemical changes, but also its optical properties may also change. Optical properties means the cell generally it is a transparent. Okay. The optical properties in the sense the refractive index, whether it will change or not, while a normal cell is transforming from the normal stage to the malignancy or some other diseases also, then not only the biochemical assay reaction will take place, but at that time also the optical properties it may vary. Similarly, the electrical properties will also vary. Electrical properties means the cell it has the membrane and inside you can have a cytoplasm. Now, the cell membrane is considered as a capacitance in a electrically equivalent capacitance. The cytoplasm it is resistive in nature, it has its own resistivity. While the disease is taking place, then this cell membrane, the capacitance of the cell membrane and the resistivity of the cytoplasm will also vary. Okay. Can I detect it? Similarly, the mechanical properties, the Young modulus, cell is a very soft, but still it has its own Young modulus properties or other properties definitely. Okay. The malleability, okay. The surface, how smooth the surface, cell membrane surface, it is we consider it is a very smooth, but it has been observed that if it is a cancerous cell, its surface roughness increases, surface roughness increases for the cancerous cell. As the surface roughness increases, so I consider that the capacitance of the cell membrane it will also increase, is it not? Because roughness increase means total surface area increases. As the total surface area increase means the capacitance will also changes. Similarly, the Young modulus or the other mechanical properties will also changes, okay, which is happening at the cellular level. How I will detect this then? So, these all are the mechanical, electrical, optical properties, I call it is a non-biological parameters. So, that will also changes or modifies during the disease progression time. So, can I detect those non-biological parameters using some devices? Okay. That is the main challenge. just think of it is happening at the cellular level, whatever changes will occur, say it is a roughness values or the refractive index or it is a bioimpedance, it is more changes occurs at a very small or the low level. How I will detect it? That is the most challenging part, because the changes occurs, it is mostly it will be merged with the noise. Can I have a probe which can detect this signal at that dimensional level? Then only you will get the proper signal. Otherwise, if I do and the cell culture on the petri dish macro device and I want to detect the roughness factor, you will not get anything. My probe should be so small that it will be at that dimension only, then only I can detect it properly. That is the main thing and important aspect that I must we should consider it. Okay. And there actually you can use this miniature technology. Okay. So, thus alternative path is explored for the level free detection of the cancer cells by identifying and measuring those non biological parameters. And here in my lectures I will mostly concentrate on the electrical signals. So, starting with that, the disease causes alters in the biochemical compositions. Biochemical compositions means it is a membrane, cytoplasm or nucleus that will alter, and it will also alter the dynamics of the cell division, adhesion and the death process. All these things will be affected. Now, the conventional technique is the labeling technique I have already discussed by fluorescence tagging. Efficiency of the techniques, it is a time consuming and it is expensive also. It is time consuming as well as expensive, because whatever biochemical assays we do, we require lot of chemicals and that is expensive, as well as it is a time consuming also. Okay. Now, coming to the other side, this also reflects in the mechanical, these changes also reflect in the mechanical, electrical and optical properties. Now, microscale detection system to probe the cellular level information during the cell culture. So, here you have a cell, this is the cell membrane and it has its own thickness. Now, cell membrane it is a lipid bilayer where it is a dielectric material, and so you have a dielectric properties which leads to some capacitance value. It is a very small value, okay, femtofarad or the picofarad range it will come, and inside you have a cytoplasm and also you have a nucleus, but in the present analysis we have neglected the nucleus because it is considered only a very small fraction of the total volume. So, we will consider only the cytoplasm, which is generally a resistive in nature, and therefore, you have the resistances. So, altogether if we consider a cell, what is the electrical equivalent? You have a resistance, you have a capacitance also. Now, we will see these things, I will come back to that slide later on. The dielectrophoretic microfluidic device for the separation of the cells. So, here how we go for that, as I told, it is with a you take the samples or the cells, do the cell culture, then you go for the fluorescence antibody antigen reaction, fluorescent tagging and see under microscope. Okay. This is the whole conventional process. So, instead of that I will use a level free technique, level free and continuous separation technique using the MEMS technology to characterize the electrical and the optical properties. Okay. Generally for the cell separation, in many of the cases while going through the biochemical assay, we have to do the cell separation. You take the samples, biopsy samples, we take it, and then in the samples, biopsy samples, you have a normal cell, also you have a cancerous cells, you have to separate. After separation you do the cell culture and then you can go for the biochemical analysis. So, how you do the cell separations? It is a microcentrifuge, fluorescence activated cell sorters, magnetic activated cell sorting or dielectrophoresis. The microcentrifuge, this is the whole thing, you can understand how you can separate it. You have a different sizes, you take the cell in a tube and then you do the centrifuge, then due to the mass it will be separated out. Okay. This is a standard technique, I am not going into the details. Fluorescence activated cell sorting. What you have here again it is a labeling technique. So, it is being labeled within this chamber, different cells are there and it is labeled with the different fluorescence tag, and it will then pass through this channel. While it is passing through this channel, a laser light is irradiated and this side you have a detector, photomultiplier tube, it will detect whether it is a which color it is. Accordingly, when it just passes through this come out from this channel, it will be tagged with the positive or negative charges. Okay. And then it will pass through this kind of a device, where this side you have a negative potential and this side you have a positive potential. So, this negative, as due to the red color, by sensing through this detector it will be charged with the negatively, and then as it comes and here and it will be attracted towards this direction. So, these cells will be move in this direction, it will be collected by this tube. If it is a green color, then it will be tagged with the positive charges, and so while passing through this it will be attracted towards this side and it will be collected this way. In this way you can separate these biomolecules. That is why it is called the fluorescence activated cell sorting. Initially you have tagged with the fluorescence colors and then the laser, through laser light irradiation you can separate these things. So, it is also a very common technique, FACS, very common technique, but here also again you can see that you have to go for the labeling process, fluorescence tagging process. Now, one of the physical process, it is known as a dielectrophoresis. We can utilize it, and using the electrical properties of the cell itself, I can separate in the and the cells. I do not have to tag with the fluorescence levels. There is no need for the antibody antigen reaction, and you can do it without that. And while in a microfluidic channel, the cells will move through that due to its electrical properties. In particular, the dielectric property of the cell can be utilized to separate the cells. Okay. So, here just I will go through some of the very basic equation and then the device fabrication, and then how we can separate it, and after separation what we will do. We have separated the cells, now I have to characterize it. For characterization we have to count how many cells are present, how many cancerous cells are present, how many normal cell is present. So, you require a counter which can be done electronic with the electronic circuits, and after counter how many cells are detected, then you can go for what is the bioimpedance properties of the separated cells, and from that the bioimpedance signal you can do the signal analysis, and you can get what is the membrane capacitance, what is the resistivity of the cytoplasm. So, at the cellular level the electrical equivalent circuit you can do it. So, what is a dielectrophoresis? The translational motion of the neutral particle caused by the polarization effect in a non-uniform electric field. Here you can see this is the one electrode, this is one electrode which is a very large area, and other side you have another electrode which is a very small. Okay. So, this is the one electrode we can consider, and here I can consider a pointed electrode. in capacitor what happen you have a both sides you have a two plates which is more or less same of the area. So, what is the electric field? If I apply a potential, what will be the electric field? Electric field between them will be very much uniform. Okay. Instead of that, to same size electrode, I can put a this kind of electrode. One is of a very broad area, another is just a single pointed tip, and apply the voltage between these two. So, how the electric field will be? Electric field distribution will be just like this. Okay. So, here I can say is the electric field is uniformly distributed? No. Here you have a uniform distribution, but as it comes towards this pointed electrode, here you have a electric field is very high. So, it is a non-uniformly distributed electric field, you can create it by this fashion. And if you have this non-uniform electric field, then I can this due the dielectric properties of the material, due to the dielectric properties of the material, it can go to the higher electric field side or the lower electric field side. Okay. Just like your paramagnetism and the ferromagnetism, one it will go to the lower magnetic field, another will go to the higher magnetic field side. Skip this, you have a various advantage of this DAP and the applications. So, most of the applications are the separation, cell focusing, particle manipulation, particle transporting, and this particle rotation, etc. Now, so you have a dipole and polarization. How dipole is created? A dipole polarization means it is a process of the alignment of charge on a body such that a positive and negative bound charges move to an electric field, resulting in the center of charge aligning along the field lines. So, you consider a particle which is actually a neutral in nature. Consider a particle which is a neutral in nature. Now, if I put the particle inside a electric field, this side you have a positive and this side you have a negative potential. So, you have a uniform electric field across this, but due to the presence of a positive and negative charges over this, the charges present in the particle will be separated. Negative charge will go towards this and positive charge will go towards this. So, although the particle is neutral in nature, but due to the presence of the electric field, the surface charges will be separated out, and for that you will get a this polarization or the dipole, P will be equals to Q into D. Q is the charge, total charges, and D is the separation between these charges. Okay. That we know this is the dipole, and this whole thing is called the polarization effect. Okay. And that happens for the neutral particle only, for the neutral particle. Okay. Now, here again I have put it, this is the positive potential and this is a negative potential, and this is the neutral particle, charge is separated here, and what happens within this you also have a media, you also have a media which is also polarizable media. Now, you think of that this media, the negative charges is happening here, and due to that the positive charge will occur here, and in this side the opposite cases will occur. Now, if the charges present in the particle is much more compared to the charges present in the media, then the total dipole effect will be in this direction. Okay. This will happen. Again I am repeating, you have a electric potential between these two electrodes, for that you have a electric field, and a neutral particle is present along with a media. In the earlier case I have shown only the particle, insulating particle. Here you have a particle, neutral particle, along with that you have a media which is also have the dipole effect. Now, if the dielectric constant of the particle is such that the separation or the charges accumulation is much more compared to the media, then the dipole effect of the particle will be much high. So, the dipole movement will be, or the vector diagram will be towards this direction along the electric field of the particle of the electric field. But in the other case, if the media has a much more dielectric properties, then here the positive charges is much more compared to the negative charges accumulating inside the particle. Therefore, the total dipole moment will be just a reverse in this direction compared to this. So, these changes are occurring due to the presence of the media and the overall effect of the dipole dielectric effect of the particle as well as the media. So, what happens, since the dipole consists of the positive and the negative charges a distance apart, they generate their own electric field. This then wrap the external electric field. So, externally you have this electric field, the uniform electric field, but due to the if we consider this case where the charges separation or the dipole is much high in the particle, then how the electric field will be distributed? The electric field will go through this particle only, because here you have a much more dielectric effect compared to the media, just like your again the paramagnetic field and the diamagnetic field. Whereas, in the other case, as if we consider this case where media has a much more dielectric constant, then the electric field will be stays in the media only, very few electric field will go through this particle. So, these changes will occur. And now coming to the some of the basic equations, how for the dielectric media where the energy losses. So, you have a resistance and you have a capacitance, and the total resistance will be equals to d by sigma into A, and C will be equals to dielectric constant into area by d, and total the impedance Z will be equals to 1 by j omega c. The complex the impedance will be given by this equation, and finally, it is the complex permittivity which will be equals to epsilon minus j into sigma by omega, and this is the real part where is energy will be stored, and in the imaginary part this component will lead to the energy loss. And now, as the complex permittivity you have a omega term. So, the omega tends to infinity means the permittivity dominated behavior it will be. If omega tends to 0, then conductivity dominated behavior it will happen. This is just a small mathematical equations which will be useful later on to analyze the bioimpedance data. Now, considering those effect, the dipole moment effect, now instead of having the uniform electric field, you can have a non-uniform electric field just like this. This is the one electrode and this is the pointed electrode, and you are applying a voltage between this electrode and this pointed electrode. So, electric field distribution is like that, and then you are putting the particle over there along with the media, not only the particle, but your media is also present, which also has some dielectric value. If in the two cases may happen, as I told earlier, in the first case, if the dielectric constant of the particle is much high than the media, then the electric field distribution will be like this. So, much of the electric field will go through the cell, through the particle, and in the other case very less number of electric fields will pass through the particle. Much of the electric field will be distributed in the media itself. So, accordingly you will the force, dielectric force will be for this particle will be going towards this direction, and in this case the particle will try to move in this direction, just in the opposite direction it will try to move. and the force. So, if you calculate total force will be equals to q into E and minus this, and finally, it will be the force for the dielectrophoresis force will be equals to this formula. Total formula is this. Here you can see that A is there, A means the radius of the particle. Okay. Epsilon m means it is the dielectric constant of the media, and the epsilon 0, then the real part of this factor and delta E square, these terms are very very important. Okay. This is the total force acting on the particle due to the dielectric phenomena. Okay. Now, here if you consider A is the radius of the particle. So, if I have a mixture of particles where the dimension is different, can I separate it in this media? You consider here in the whole thing, it is merged in a liquid. Inside the liquid you have a one electrode which is broad and another pointed electrode, and then you have a number of particles, dielectric particles, where dimensions will vary. You have a larger particle, you have a smaller particle also, and all particles are neutral in nature, Okay. Now, can I separate this by using this electric field? Using the electric field, can I separate these things, these particles? It is possible, because the dielectric force, this is the equation for the dielectric force. If we consider these things are constant, then you have a variation of A. That means, you have a larger particle, you have a smaller particle. For the different particles, you have a different forces will be acting, and accordingly as the forces are different, they will move in a different direction. Okay. So, by that way, by the size distribution, you can separate the particle. Okay. That is the one aspect. The second aspect is that if the size is same, then if but your dielectric constant of the different particle is different, varies, then also you can separate the particle, because here this force is dependent on this one as well as this factor, where you have a dielectric constant. Okay. I utilize this phenomena, this dielectric variation of the dielectric constant of the force, that happens because of the propagation of the diseases. Due to the disease, a normal cell will have a different dielectric constant compared to the malignancy cell. Okay. As the disease progresses, the dielectric constant of the cell will changes. What is the dielectric constant? It is the dielectric constant of the cell, which comprises from the cell membrane capacitance as well as the cytoplasmic resistance. When the normal cell will transforms to the disease cell, its the resistivity properties, the capacitance properties will changes, and that leads to the change in the dielectric properties. Okay. So, although both the cells are same dimension, okay, you take if you take one cell, it is a normal cell, it has its own dimension, particular radius. When it transforms to the malignancy, the dimension will remain same, A value will remain same, but the dielectric constant changes. So, I utilize that properties for the separation of this disease cell and the normal cell using a microfluidic devices. That is the whole concept, and besides this A and the dielectric constant, you also have this one. What is that? E is the electric field. So, the gradient of the electric field. So, I do not require the here for the separation to get the dielectrophoretic force. I should not have the uniform electric field, I should have a variation in the electric field. Okay. And that is why you require a non-uniform electric field. from this formula itself I have to have this kind of thing, and therefore, I require a non-uniform electric field, and to get the non-uniform electric field it is very simple. One electrode will be broader, another electrode will be very pointed. So, that you will have this kind of distribution. Now, come to our thing, let us just for some slides I have given how to understand what is the physics behind that, and now come to the actual devices. Okay, some more things. Okay, this one I will just discuss. Positive and negative DP. Here, so my main aim is to separate the cancerous cell from the normal cell. You take the biopsy sample, and then there you have a mixture of cells, that means mixture of normal cell and cancerous cell. Both the cells have the same dimensions because its origin is same, but their dielectric properties has differed because the disease progression has taken place. Okay. I will utilize this dielectric property to separate the cell. How will do? I will take a microfluidic channel, and inside the channel I will flow the cell mixture of cells from one side using some media. Its cell cannot be just I have to keep it in a media with the nutrients and all, and it will flow through the microfluidic channel. While it is flowing, one by one cell will flow through the microfluidic channel. I will put a electric field, non-uniform electric field, using some electrode within the channel. Okay. So, when the say one cell will pass through that, due to the presence of electric field, that dielectrophoretic forces will be experienced, and the force, how much will be the force or what is the direction of the force? It will depend on what is the dielectric constant of the particle as well as what is the dielectric constant of the media, because both are inside in the equation. If the dielectric constant of the media is much higher than the epsilon value of the particle, then this particle will move into the one direction, that is known as a negative DEP. Dielectric constant of the media is much more than the dielectric constant of the particle, that will be decided by how much charge distribution is there. Okay. So, accordingly for this case the particle will move towards lower electric field side. Lower electric field means this side you have a lower electric field distribution. So, particle will move towards this direction, whereas if the dielectric constant of the particle is much higher than the epsilon value of the media, then this particle will experience a force towards this direction, that means where you have the maximum electric field distribution. For this case it is going towards the lower electric field distribution, for this case it is going towards the higher electric field distribution, and same has been shown here. The red color means the dielectric constant of the particle is much higher than the media. So, this red particle is moving towards this direction where you have a maximum electric field distribution. For this particle it is the dielectric constant is much lower than the media, and so it is moving towards this distribution electrode size. Okay. This is the total physical concept, and for your information the whole thing, the f value, if you consider here this f, it only depends on this the resistivity as well as the frequency. This omega means it is the frequency of the applied electric field. Okay. So, these are the different parameters which you can play. Yeah. And again you have equations by which we can calculate what is the actually the dielectric force. Okay. I will move to this, and non-biological area, DNA. Yeah. Now, come to this your fabrication part. So, one way I can have a electrode like this, this is a planar electrode which are pointed to each other. If I apply a voltage, the electric field distribution will be like this. So, maximum electric field will be here and the minimum electric field will be in this within this region, okay, but maximum electric field will be here. When the particle will flow through this and the electric field is applied, the positive dP particle will be accumulated or trapped at this region, whereas the negative dP particle will be trapped in this region. Okay. So, this is the one way you can get the separation, and how this is happening in a microfluidic flow. So, you have a microfluidic flow, you have a electrodes and the cells is moving through that, you apply the voltage and then the cells will be trapped at this region and other cells will be trapped in this region. After that you close the input and start the your supply of the buffer liquid. As the buffer liquid will flow, the electric field distribution is also stopped, then these cells will be first removed and you can collect it, and after that you can collect with a higher buffer flow, you can collect these particles which are trapped along the different corners of the devices. That way you can separate, you can get the separated cells, but it is a sequential process rather I will say, because first you will supply the samples, apply the electric field, it will be trapped, and then after that you will switch off the input flow, switch off the electric field, then apply the buffer and get the separated cells. It is a sequential process. Instead of that, I can have a continuous flow and continuous separation. Some of the other electric fields it is shown here. So, how will get the continuous the separation of the samples? Instead of having this kind of a electric field, I can have a electric electrodes like this. It is a total planar view. Basically, you can take a glass substrate, starting with a glass substrate. This is a glass substrate. On top of that, I can have a metal electrodes that the configuration is like this. This is the one broader electrode, and these are the two small electrode going in this direction at a particular angle. And
what is this electrode
made of the gold generally.
And they can go with all the mammalian cells.
Because gold is a noble material. Otherwise, if you can use the any metal can be useful to get the electrode structure, but as for the biological samples we have to use the different media, and that media will react with the different metal. That is why gold, as gold is a noble material, you have to go for the gold electrode. Okay. So, this is the design, and then at the inlet side, this is the inlet side where you have a 3 channels. One is this channel, this is the media second one, and this is the another one. The samples will flow through this, and 2 buffer solution will flow from these 2 region. And on the other side you have a output 3 outlet. These are the 2 outlets at the 2 extreme corners, and this is the middle outlet. Okay. And if you see the cross sectional view along this direction, it will look like this. So, this is the glass substrate, and here you have a 1 electrode, and these 2 are the finer electrodes, and this is the middle electrode. It will look like this. So, this is the total planar view, but on top of that you have to make some arrangement for the channel. It should be covered with this, so that the fluid can flow through that. Okay. So, this is basically a microfluidic device integrated with the electrodes, and you have a different channels at the input, and also at the output you have the different channels. Okay. What is the dimension, how much will be the angle, this can be calculated from this. Okay. This is the main equation, and from that you can have what is the length and of the device, and what is the angle to be made, so that at a particular distance, after travelling a particular distance, the cell will be separated and I can collect it from here. Okay. So, this is the design aspect. Now, how much is the just one or two slides I have for the simulation before going for the fabrication. What is the electric field distribution? Electric field here, these are the two main electrodes and this is the common electrode for the two. Okay. So, it will be grounded and these two electrodes are the active electrodes. So, here the simulation result shows that the electric field will be confined around this region and also around this region where you have those two electrode structure, and in at the middle portion you have a 0 electric field. Now, okay, I will skip this one. This is the fabrication. So, you start with a silicon wafer. We have started with a silicon wafer, but it can be a glass slides also used as a substrate. So, take the silicon wafer, grow a silicon dioxide layer. On top of that, you can go for a chromium and gold thin layer of deposition. After deposition, you go for a lithography, and using a mask you can pattern these electrodes like this. Okay. After fabrication of the electrode, you have to make a channel over this to get the microfluidic channel. So, for that you have to have a second lithography. So, coat it with another photoresist, then do the again the lithography and the ultraviolet light exposure and make two pillars at these two corners, and then you can cover it with some polymer layer which is known as a PDMS, polydimethyl siloxane. This is one kind of a polymer. So, that will be covered to these pillars. So, now this is the complete fabrication process comprising the two lithography step. So, after this what you have? You have one channel, covered channel, inside that you have a electrodes, integrated electrodes, and also one side you have a input, another side you have a output channel. Here probably picture is not very much clear, but today we have a lab visit and you will see those devices also. So, here very you can say this is the actual device is like this, only a few millimeter in length, only 5 to 6 millimeter in length, total length of the device. Okay. And at the input sorry, this yellow color, it is the gold electrode you can see, and this is the larger this is the magnified view, and here you can see this is the middle electrode and these are the side electrodes. And these electrodes has to be connected to the external power supply. For that you require a bond pad, as it is done in the ICG. Okay. And that is at the periphery, bond pad is at the periphery. And then on top of that you have a microfluidic channel which is transparent in nature, but here you cannot, only you can see this is the boundary line of this microfluidic channel. Okay. And it is a totally transparent thing. Now, at the input side, this is the input, one input I have shown here, this is the one input, and this a pipe is arranged to this and to the reservoir of the medium. And at the output, this is the output where you have these three channels, three reservoirs where the cells, separated cells will be collected. Now, we have to do the testing. After fabrication we have to go for the testing purpose, and here it is like this, the total microfluidic device, and how the cell will be injected, it is by a syringe pump. A syringe is connected with a pump which is a computer controlled, and you can flow as much as 1 milliliter to as low as 1 picoliter volume. Okay. So, it is called the syringe pump, is connected to the inlet. So, the cell along with the media, mixed cell, normal and cancerous cell can be flow through this syringe, and then it will come and it will flow through this microfluidic device, and then you can apply the electric field at these electrodes, and then you can collect it. The outlet is connected to some 3 petri dish, that is all, and we can observe it through using some microscope. I will quickly read out what is the result. Here I have started with a polystyrene bead. My aim is to separate the cancer cell from the normal cell, but I started with a polystyrene bead. Polystyrene bead is a insulating bead having a dimension or diameter of 1 micron, 10 micron, like that. It is available in the market. Okay. It is a insulating, a small particle, round shape particle only. Okay. So, I started with that, and it has been observed that this polystyrene bead also has the dielectric constant, epsilon value is there. Okay. The epsilon value and it is mixed in some media. The epsilon value of the polystyrene bead is much higher than the media, and we have, so while it is flowing through this, while it is flowing through that, we have changed the frequency of the input signal from 100 hertz to the 10 to the power 7 or 1 megahertz, more than 1 megahertz. Okay. We have changed the input frequency, but nowhere we have found that in almost all cases up to megahertz the particle is flowing in this direction, straight away it is going from inlet to the outlet channel. Okay. If there is a positive DP, positive dielectric force, then after injecting into the input, due to the positive dielectric force, the particle will move in this along this direction, or it will move in this direction, in this way it will go. But as it experiences a negative dielectric force, because of this dielectric value, it moves along this straight way, it goes in a straight way and collected at the center of the reservoir, central reservoir. Okay. Because its dielectric constant is very high, and within this frequency range we cannot get the positive DAP force. Okay. But anyway, we have to, at least come to this conclusion. Next we have started with the Hacker cell. Hacker cell is a standard epithelial cell, and we have cultured it, and then we have mixed with the media at some particular concentration, and then we flow through this, and we have observed that at 100 kilohertz to 600 kilohertz frequency range, Hacker cell experience a negative DAP. Okay. Negative DAP means it is flowing along this central line, there is no deviation, it is coming inside the channel and going straight away towards the outside outlet. Okay. But when the frequency changed to more than 600, from 800 kilohertz to 1 megahertz range, then we have observed the positive DAP force, and Hacker cell was observed that it has gone towards this direction, very few number of cell available at this. Whereas at the low frequency, at it is mostly the negative DAP force, it is going in this direction, whereas at the higher frequency it moves into this direction and going in that way. Okay. This we have observed, this is the experimental result, and here in the theory what we have observed that at 736 kilohertz, where it is the crossover frequency. Crossover frequency means this force will change over from negative to positive DAP, that is the crossover frequency, and that we have observed theoretically, and that is why this at the lower frequency it is negative, but around 800 kilohertz onwards it is going to the positive DAP force. Okay. And the next is your microbeads and the cells. Now we have mixed up two things, one is the biological cell, another is the polystyrene bead. Polystyrene bead we have observed for the whole range up to 1 megahertz it is a negative, whereas at the this Hacker cell it shows about 800 kilohertz, it shows the positive DAP. So, we have mixed up these two things, okay, and flow simultaneously through these channels, and we have observed that at the 100 kilohertz most of the cells and the polystyrene beads are going through the central of the line, because it both will experience the negative DAP, whereas above the 800 or 1 megahertz it has been shown that the cells are going to around the edges, whereas the polystyrene bead is moving in this direction. So, what you can get we can collect at the input you have a mixture, but at the outlet it will be separated and you can collect it separately at the different chambers. Okay, I will stop over here, and after T I will go through that what you can do after this. I told that my aim is to separate the cells, then I will count how many cells are present, how many normal cells and how many disease cells are present in the sample. After that we will go for the bioimpedance study to characterize the cell properties. Okay. So, separation has been done using that DAP force and the microfluidic device. Then I will discuss how we can go for counting without labeling process. Please remember that, okay, because most of the biological samples, biochemical assays we do with the labeling, with the fluorescence tag and the antibody antigen reaction, but here we have started in such a way that there is no antibody antigen reaction and no requirement of the fluorescence tag. Using the electrical properties you can separate and then do the we will go for the counting. Okay. So, let us have a break, and after 10 minutes we will start again. So, this like this manipulation of the nanotubes. Okay. You can make a nanotubes, and then if I want to align it, all the nanotubes should be aligned at one particular directions. Then I can go for this kind of they put a drop of this nanotube and then use a electric source, and by putting the giving a electric field, all the nanotubes will be aligned only in the one direction. That is one just a applications, okay. And it is useful for the sensing the gas, humidity, molecule, etc. And this is the one ACM photograph where it is not ours, have taken it from one paper. One nanotube, I have to characterize one nanotube. Characterize means electrical characterization I have to make of a say carbon nanotube, xenon nanotube that we have fabricated, characterized by IV, current voltage characterization. Okay. How will do? You require the two pads, just like a resistor you require two sides, the two connection, but nanotube it is a nanometer dimension. How will get these two? So, basically it is a you can make the two bond pad. This is the two bond pad at the two different locations. Okay. That you can do using a lithography process. After doing that, you just drop cast a nanotube by liquid drop cast within this region, okay. And then so nanotube will be placed within that between the two bond pads. Then I can apply the electric field between these two electrode, it is already there, and the electrodes are made in a such a way that it will be a non-uniform electric field can be generated, and then you drop cast that whole part where you have a where you have this nanotube at a certain concentration and apply a electric field between these two. So, it will be aligned between this and it will be connected between this point to this point. Now, with a see under ACM, see under a high power microscope, you can see whether the nanotube is connected between these two or not, or how many nanotubes are connected. Here I have shown only the one, how many nanotubes are connected here in this local region, and then I can go for the IV characteristics of the nanotube. Okay. So, by this is the one way people are doing utilizing the dielectric property. The third one is the cellular concentration and the biopatterning. So, I want to make some biopattern on some substrate, any pattern I make it. So, here one kind of a cells are there, different applications. So, just put this your samples and go for the electric field, and then you will get this kind of a pattern. Several applications are there. Okay. So, this is one of the technique, or it is a tools which can be used to pattern or the either a biological sample or non-biological sample, and then you can characterize it. Okay. Now, coming to our part, the bioimpedance based microflow cytometer means after that separation I want to count how many cells are present, how many cells are coming over there. Okay. So, counting the number of cells or particles in a fixed volume of sample. So, again this is the microfluidic things, the different techniques, different advantage, disadvantage. Now, what we do here, you can remember that in the dielectrophoretic microfluidics channel inlet, the flow the particles will flow through this electrode and giving the electric field, and based on the dielectric properties of the media and the cell say and the frequency of the electric field, it will be separated out. And while after separation, as it is moving towards the outlet, I can place two electrodes like this, which is very small. And 2 electrode means just the 2 electrodes which are very nearer, close, only 10 micrometer apart or 5 micrometer apart, 2 electrodes. Again it is made of the gold material or any such kind of a material which is sustains in the media. Now what happens, and this is the one common electrode I have made, and this is the individual electrode that is there. Okay. So, series of electrodes are placed along the channel. Now, and these 2 electrodes are connected to the voltmeter. As such, in general, the fluid is flowing through that, fluid is flowing through this channel. Okay. So, based on the dielectric property of the media, you will get a voltage across these 2 electrode, common electrode or any one electrode. Okay. But along with the media, sometimes because concentration of the cells is very less, sometimes one particle is flows, either it is a particle or it can be a cell also. So, what will happen to the voltage? Voltage will changes. In its continuously flow of fluid is there. So, accordingly you have a voltage. Now, instead of a fluid, in addition to the fluid you have some cell or some polystyrene bead is passing. So, depends on the dielectric property of that particle, that voltage, that signal will changes. Okay. If I detect it, if I detect that signal through some circuit, okay, and then count that those number of signals, that will give you how many cells are passing through the channel. This is the very simple, because the counter we have in the electronic circuit, you have standard circuits for that. So, here it is basically those 2 electrodes are placed with the circuit, connected to the circuit, and a voltage shift of the voltage level will be there. Shift of voltage level, because all the times flow will be there, fluid flow will be there, for that you have a voltage scale, voltage level, but along with the fluid, once one particle will move. So, you will get instantaneously a shift of the voltage, okay, instantaneously for a short duration. How much is the duration? That will depends on what is the cell dimension and what is the flow velocity, okay, because the cell, this dimension of the electrode is only a few microns, gap is only 5 to 10 micron, and dimension, lateral dimension only of the order of cell, and dimension of the cell is about 10 to 20 micron. So, as it moves, crosses over that, voltage level changes, and you can easily detect it using a standard electronic circuit, and that will give you a counting of one number, and that way you can count how many number of cells are across that. Okay. So, that way you can get the number of cells, how many number of cells there for the cancer, how many number of cells are there for the normal or disease, that way. Secondly, as there is a shift in voltage, so the shape of the voltage signal, from that you can get what is the dimension of the cell, because it will depends on what is the velocity, what is the electrode dimension, and what is the cell dimension. Based on that you will get the signal level shift. So, analyzing that signal, you can get the characteristic features of the cells also, cells or the particle also, both the information you can achieve. And so, here we have used this kind of a circuit, it is a very simple circuit, and using a DAC we have tried to capture the signals or count the signals. Okay. So, this is again the cytometers, the FEM modeling you can do. and this is the electric field distribution. This is the two pair of electrodes are there, series of pair of electrode. When the fluid flow is there, this is the actual device and this is the simulation, and for the fluid flow what is the electric field distribution, and here you can say the impedance variation in this curve you can see this. So, this is all about the your simulation. So, one particle it is moving through that through this electrode, and how the electric field variation is creating, it has been shown here. Okay. This is the electrical output. Yeah. Now, in the actual device if you see, so here you have a electrodes sorry DP for DP your device, and after that you have this array of electrodes, the enlarged version is shown here. Okay. This is the common electrode and we have a 5 number of serial electrode. You can capture the signal once this one will be passed through that, then this accordingly will get the signals. Okay. A series of electrode we have taken because sometimes one bubble may pass through that also. So, how to say either it is a for the bubble also you will there will be a shift of the shift in the signal. So, how you say it is a bubble or it is a cell, for that you have to go for analyze you have to have a different series of the electrodes to confirm yes whether it is a bubble or it is a different thing for that. Okay. And also we have some modified instrumentation part also, where we have used the notch filter 50 hertz and the comparator all these things, and it will directly go to this pulse counter and display the how many particles are present, or it can be captured through this DAQ. Okay. So, two different ways we have done it, and this is the final circuit part, and here I can say the display you can see the how many particles are have come crossed through that channel. Okay. And this is some of the results that you see particle or cell concentration we have taken about 10 to the power 8 bits in 2 ml PBS solution. Okay. And flow rate was about 2 microliter per minute. Flow time is 40 seconds or 60 seconds. The real time count is this, real time means here, and through the DAC it was like this. So, it was more or less the similar result we have found with the real time count and the through the DAC. For the 10 to the power 6 cells in 2 microliter, then the flow rate is about 2 microliter per minute, and the number of particle that has that crosses this is like this. So, more or less it gives similar results, and so this way we can establish that using a microfluidic channel how many particles are crossing, that also can be measured. Okay. Now, once you have separated, then I have counted how many cells have passed through that. You can estimate, yes, you have a so many number of normal cells, so many number of disease cells, you can have this. And next is the you have to characterize these cells, characterization, and basically what we did, it is a electrical characterization. Okay. You can go for the other characterization also, but here it is mostly the bioimpedance study we have done it here. So, and different aspect of the bioimpedance study that we will now discuss. So, basically the imperometric assessment of the cellular behavior means what is the change in the R, C, epsilon and rho value. So, you have a resistance corresponding to the cytoplasmic resistivity, you have a capacitance value, capacitance comes from the cell membrane which is basically a dielectric property, and for the imperometric study two types are there. One is the frequency sweep at a fixed time, and another one is the real time at a fixed frequency. Some of you are aware that impedance, okay, it is in the electrical domain where you have a capacitance, resistance, inductance. change of and it will creates a barrier to the current flow. Now, for the cell we consider it as a particle and you have a combination of the capacitance and resistance. Capacitance comes from that your cell membrane and the resistance comes from the cell cytoplasmic resistance. Okay. So, as the cells are now separated out, we can collect it in a petri dish, in a miniature version of a petri dish, and you can go for the impedance study. And there you will get some impedance signal, you will get some signal, okay, because bioimpedance is a very old subject. People use it is more than 100 or 150 years old. We have to use if I put the two electrodes here and here, I will get the impedance, something, some results I will get, because as the current flows based on the properties of the tissue and the other pathway, you will get the different values. Okay. So, it is a old study, and from that you will get you can get different characteristics, properties of the tissue and the other organs, so many things are there. Here we want to study at the cellular level, where the current will pass through the single layer of a cells, and it is within a very confined region, so that I can capture the signal which is very weak. I am not doing it in the large scale or a conventional scale. I want to do it in a miniature version, so that I can capture the smallest possible signals. I can capture it and I can analyze it. Okay. If I want to capture the signal at a cellular level and using a conventional macro device, you will not get the signal, it will be merged with the noise, as simple as that. So, I have to put it in a micro environment, that is the main aim for this. So, but to start with, and as again we have the engineering background students. So, just a few slides I will put over here to discuss just as simple as the how the cell culture is being done. On the other side, for the biology background students, what is the impedance. For impedance you can do it in a two different way. One is the frequency sweep at a fixed time, another one is the real time at a fixed frequency. That means, first of all, for to measure the impedance, you require the two electrode. Again, between the two electrode you will put your sample, whether it is a wood, it is a paper, it is a cell, put the samples over that, and then you pass the current and I mean the AC current, and you can change the frequency of your signal, okay, input signal, and accordingly you will get the Z value. Z means it is comprising of the C and R. Here I am not considering L factor, inductor I am not considering, I have only C and R. Now, if at a particular at a fixed time, if I sweep the frequency from low frequency to high frequency, then the characteristics impedance will change. That change will occur due to the change in the frequency, because the resistance it will not change with the frequency, but the capacitance value, as it is it depends on the frequency. So, its value changes, and so overall impedance will change. Okay. Just for example, if I take a cell, again it is a cytoplasm which is capacitive in nature, and inside you have a cell capacitance, cell membrane which is capacitive, inside you have a cytoplasm, and I am putting one cell between the two electrode. Just consider I am putting one cell between the two electrodes, and I am giving a AC signal. What will happen to the impedance? How much current will flow through that? DC current I have given. So, as it is a capacitive nature, the cell membrane, current will not be allowed to flow through the cell. It will just pass from the outside, it will pass from one electrode to another electrode. cell it is sitting on the electrode sitting means it is within the media itself, within the media. So, current will pass through the media, but I want to capture the characteristics of the cell using the AC signal, but AC signal is not allowed to pass through the cell. I will not get any characteristics of the cell, I will get the characteristics of the media only. Now, you change your increase the frequency. So, slowly and slowly that capacitive nature of the membrane will be lower, the reactance of the membrane capacitance will be lower and lower, and the it will allow the current to pass through the cell. Okay. And so, as it passes through the cells, so inside you have a cytoplasm and all, the final Z value will be changes. Okay. So, based on that you have this frequency sweep. With the frequency, if you plot the impedance value at the 0 frequency, you will have a very high impedance, but as the frequency is lower and lower, the reactance will be lower, frequency higher and higher, reactance will be lower, and the total impedance value will be lower. So, this is the general characteristics for the frequency sweep for the cell. Okay. On the other side, for a real time at a fixed frequency, that we have done during the cell culture time, Okay. What is now the cell culture? Coming to that engineering student, that you take a petri dish and take a small amount of a cells, any cell, generally the cells if I want to use my cell, I cannot use it. For your information, I have to go for what? Cell lines, because if I want to use my cell, then I have to go to the ethical committee's clearance, which is really very tough to get. So many protocols are there, I mean things are there. So, we have to go through that, then only I can use it. Animal cells I can use, for that also you have to go for the ethical committee clearance and all. So, but at the same time you have a different cell lines are available in the market. I can purchase those cell lines and I can use it for my experiment, and for that you do not require any ethical committee's clearance. Okay. So, I have shown it is a Hackett cell, HeLa cell, different cancer cell lines, these are available, and we call it is a cell line, and I can purchase and I can directly use it. Okay. So, I take that one cell line, take a very small concentration of that and take the media means the nutrients. So, for particular cell line you have particular media, I have to use it and put it on the in the petri dish. Okay. About 10 to the power 3 to 10 to the power 4 cells can be taken in a petri dish and put it at 37 degrees centigrade in a CO2 incubator for the 3, 4, 5 days. What will happen? Cell will proliferate. Okay. It is a cell growth process. So, from 10 to the power 3 it will go divide and it will start it increasing, number will increase. Okay. It is a total growth process will take place, and ultimately after 3, 4, 5 days you will see the whole petri dish is covered with the cell lines, with the cells. Okay. And thereafter you see the cell will die out, although nutrient is present, because there is no further space to proliferate. So, cell will start dying out. This is the whole total cell culture process: growth, proliferation, and then death. Takes 3 to 5 days time. While doing the cell culture, can I measure the impedance? For that I will do real time at a fixed frequency. Here it is a frequency sweep at a fixed time, What I will get it here from this data, I can get the membrane capacitance and cytoplasmic resistance from the basic equations probably I have shown earlier slides. Okay. From that this impedance data you can get it. Okay. Here what will happen, as the cells I have incubated the cells in the petri dish, and below that I have kept the 2 electrode only. It is the requirement for the impedance study. Okay. And over that you do the cell culture. When it started at the so at the time passes out, we will do it at a particular frequency, and that particular frequency, what will be the particular frequency? Because here you can see from the low frequency to high frequency, this is the change, okay, occurring. What frequency I will choose for this? Because I want to get the properties of the cell. So, that means the current should pass through the cell, then only. But what is the low frequency? I cannot use it because it will not go through the cell. High frequency I can go with the very high frequency, no, it is about 10 to the power 4, say from 1 kilohertz to 40 kilohertz range. It is the best choice, so that the electric field will be distributed, same amount of electric field will be distributed inside the field, inside the cell, as well as inside in the media, uniformly distributed. Okay. That is why it is about 10 to 40 kilohertz. We did it at 40 kilohertz range that way. So, when it started at that time, the number of cells is very small. So, impedance is less. Now, as the cell will growth takes place, so immediately the number of cells will increase. So, impedance value will also increase. Impedance value will also increase, and then it is the cell seeding, this area it is the cell seeding process. Then you have a growth process, and then it will proliferate. Proliferate means it will go around the whole space of the petri dish. So, more or less within this time the impedance will remain constant and the confluence state, and after that if there is no space, then the cell death process starts occurring, and therefore, the impedance will start decreasing. Okay. This is happening within this cell culture. So, this is the real time at a fixed frequency, whole curve. So, you start from the day 1 and measure the impedance, and after 5 days you will get this kind of variation in the impedance, whole thing. From the real time cell growth, you can get the attachment, spreading and death process, whereas from this side you will get the cell membrane capacitance value and the cytoplasmic resistance value. So, combine the static and the dynamic features infers the complete pathological state of the cells you can evaluate. So, basically instead of this one petri dish, I can have a microfabrication process, and you can get a array of cell well, the cell culture well inside the below which you have the electrodes also. This is the complete picture of the miniature version of your cell culture dish along with the bioimpedance study. Okay. So, basically you have a 2 electrodes, I told you here. These are the electrodes you can fabricate on the glass wafers, then you can have a thin layer of dielectric. On top of that you just have a well kind of cylinder, cloning cylinder, put it together you will get it, and this side you have all the electrical connections, so that it can be directly connected to the instrument. Okay. Now, after fabrication you just dip the put the cells along with the media in this cell culture and put it in the incubator, and you can connect it to the impedance meter also. Now, cell culture will take place and you can get the reading of the impedance, but it is now for your information. We did it with the wiring. So, incubator it is just a small equipment, and from that we have to take the wires connected to the impedance meter and all, but today it is possible that the whole thing, it can be just this device can be have some small RF transmitter receiver also. Okay. So that wirelessly you can get the all the impedance data in my laptop. It is possible. Whole thing today, that kind of incubator with the bioimpedance study it is now available. Okay.
So, is this cloning cylinder acting like PDMS in that
yeah, it is a pair, yeah, centimeter to millimeters. The high, this dimension whatever we did probably about 500 micron, that means 0.5 millimeter. Instead of this much big petri dish, you can have a 0.5 millimeter petri dish, and height is only 100 micron, height of the cylinder, it is only 100 micron. Okay. So, again it is the same thing, the optimization frequency, you can go for the different COMSOL simulations, impedance study. And while doing this impedance study, one thing we have to remember that we have to get the baseline correction. Anything between the two electrodes, if I measure impedance, you will get it. Okay, that is why before seeding the cells, you have to put the media and get the impedance value, which will act as a baseline, and then you put your cells and the impedance will changes, and that is the difference between these two, you will have the impedance of the cells only. Okay, so it is the electrical equivalent, as I told from here that yeah, from this frequency sweep you can get directly the electrical equivalence of the membrane capacitance and the cytoplasm resistance. So, some simulation results, this is the electrical equivalence where you can see that cells, this is the electrodes, on top the cells has been seeded, and you will get the frequency sweep. And then what is the electrical equivalent circuit? This side you have the some resistance, double layer capacitance, and the coating capacitance coming because of the presence of the dielectric layer. This is capacitance due to the double layer. Some of you have asked about the double layer things. So, here in this case we have considered the double layer capacitance, and the resistance of the media, resistance of the solution here, and the resistance is the exposed electrode resistance we have considered. Now, this is the main part where the cells, where electrical equivalent circuit has been created for the capacitance comes from the cell membrane and the cytoplasmic resistance. So, this is the total electrical equivalent for circuit for the whole system, comprising the cells, comprising the media as well as the electrostructure. Media, for media you will have only the solution resistance, whereas for the electrodes you have as you have a electrode means it is a metal. So, you have a resistance value, you have a coating capacitance value, sorry, the capacitance for the coating layer, as well as you have a capacitance for the double layer. Okay. Using this, so it will become some more complex, but using this circuit you can get this the cytoplasmic resistance and the membrane capacitance value.
Sir, when you are giving this high frequency electrical impulse for a longer duration, like for hour basis, so whether it is giving us a single stretch, it is continuously connected, or it is
No, yeah, one this is an important question. How much voltage I can give? Because for the impedance study I have to give a voltage so that you will get the impedance. How much voltage I will give?
so
Because it is a living cell.
yeah,
What is the expected value? Is it 1 volt, 5 volt, 10 volt? It will die, simply cell will die. I cannot use that much high voltage. It is limited to 10 millivolt to 50 millivolt.
and that will giving us a
That is millivolt or microvolt, sorry, it is I actually forget that, I will tell you. This is actually I am presenting one of my students PhD work from dielectrophoresis process to this impedance study.
And that too as a giving us a continuous way, or in between you are giving some breaks or something
For the frequency sweep you have to give continuously, and there this time is only say few minutes, only 1-2 minutes, you will get the give the frequency sweep and get the data. For the real time, real time what we did, we cell culture, we have put it in a incubator. Now just say initially we started with a half an hour duration measurement. After half an hour I will give this, I switch on the impedance meter.
So
You say, but wherever you have a changeover, there I have to do it more frequently. Say maybe not below 15 minutes, not below 15 minutes. And where you have a stabilized things, it is 1 hour duration gap you can give it. And voltage is a very important, and it is 10 to 50 millivolt, na? Millivolt range. Okay.
What is the double layer?
And there up to 50 or up to 100 millivolt, cell will not die, that is ensured. Okay. But if you go beyond that 200 millivolt onwards, that electrophoresis will start. Electrophoresis is another area, some of you are aware, and that also can be used done by using the MEMS device. Okay. But
the cell disruption starts there.
yeah, and long time exposure is not at all possible. But 50 volt is a very much comfortable region, cell will not die. Yeah.
This double layer thing, so what is a double layer?
That comes in the microfluidics when you have a cell, solid boundary, you have a liquid, and also you have a movable ions, mobile ions are present in a liquid, and the surface of the solid boundary have some charges are fixed. So, this charge from the liquid, it will come and fix to that point. So, it cannot move further. So, it creates a double layer. So, it is a very basic kind, but you have a lot more things in the double layer. Okay.
I mean,
In the microfluidics, it is a very important factor, electrical double layer.
I was thinking like there we have a polymer coating and gold electrode there. So I thought like the interface between those two is acting like the double layer.
No, not there. For the polymer coating, you have this capacitance value,
Okay.
and wherever a polymer coating is not there, there you have the exposed electrode resistance, and this comes out in a different way.
So then the fluid
Yeah,
is flowing.
flowing, and you have a movable ions then, which will be trapped at the wall. Okay, and that creates a electrical double layer. So, this is all the extracting the single cell parameter from the impedance data. I am not going into that details, most of the part is a theoretical part. Only thing you get the frequency response of the Z value and put it in these equations and get the results of the single cell parameter. Okay. The result shows that this is the HeLa cell and this is the Hacker cell. Hacker cell is a normal and HeLa cell line is a cancerous cell line. What we observe, the single cell capacitance membrane, if you see for the Hacker cell it is about 50, whereas for the HeLa cell it is just about double. Okay. That observation we have made it. And also from the ACM image you can see the Hacker cell, you have a, this is the just a ACM image, so some features, some surface I have can see, and here you have a the Hacker cell which is much more rough than the Hacker cell. Okay. As the rough surface and surface is many folds are there in the surface. So, overall area of the surfaces increases. As the overall surface area increases, so the capacitance value also increases for the HeLa cell. That is our observation. So, enhanced surface roughness and the effective surface area that increases. Now, coming to the real time kinetic behavior. So, instead of a frequency response, now time sweep, impedance measurement is a time sweep, we can get it. And here this is the two cells I have shown, one is the MCF7 and another is the MDA MB231. These two are the breast cancer cell line, one is the MCF7, but this is much more malignant compared to MCF7. Okay. But here you can see that with the time variation, if you do the this is the growth, then you have a proliferation, and then you have a cell death process, and this is the impedance value, and real time impedance measurement. Okay. Here this is the one it is the more prominent. Three cell lines, one is the normal cell line Hackard, MCF7, and this is the another one. These two are the cancer cell line, but this one is the much more your malignant one. What we observe that the pattern of the impedance behavior along the time, it is more or less same. Initially there is a growth phase, impedance increases due to the growth phase, then there will be a proliferations, and the impedance more or less remains same, and there is a death phase. Same thing happens for the three different cell lines. But if we observe here the rate of the impedance growth, that means the number of cells growth of number cell growth process, it is much less compared to this one or this one, because this is a normal cell and this is the cancer cell, and cancer cell growth process is very high compared to normal, and therefore the slope of the curve at the initial phase, it is very less compared to these two, that we have observed under the impedance study. The second one is that the fluctuations, bioimpedance means you will have the fluctuations of the data, it is obvious. Okay. But here if you see the fluctuation is very less compared to this and this. Why? Because this is a normal cell, and as the cell growth process taking place, normal cell means it will growth process is taking place, but it will remain there, more or less it will remain there. But cancer cell, it is a have a very high energy, and it will try to go elsewhere, always have some and therefore it resulting in a fluctuation of the impedance. Okay. And that also we observe that for this case that is much more malignant. So, here the fluctuation is much more compared to this one. So, this initial, or I will say the initial observation we have studied using the bioimpedance. Now, the more importantly that these are the signals that we observed at the micro or the nanovolt scale region, and also using miniaturized devices. If we try to do it at a macro scale, probably we will miss these signals, because this will be merged with the noise also. It will be very difficult to get it. But whatever the electrode dimensions, it was only within the 100 to 200 micron gap. In a very small confinement, we did the experiment and we captured this kind of a signals. Okay. Yeah, here we have seen that at 40 kilohertz 5 minutes interval time has been given for this study. Okay. The other one is the imperimetric monitoring of the cytotoxic response of the cells. So, the impedance study, what we did with the frequency response, you can get the electrical equivalent parameters for a single cell or a cluster of cell, both it is possible. Then for the real time, if you do the impedance study, then you can get how the cell growth process is taking place, and then for the cancerous cell how much it is, and also we have extended that analysis further to wavelet based transformation technique to get how much energy it has. Okay, and it has been observed that, because if you are interested, you can see it is a physical review E. We have correlated this with the cellular micromotion, because cancerous cell will always migrate, and therefore not only the fluctuations, you will see the total energy of the system is also very much high compared to the Hacker cell. which is normal in nature. Okay. So, that reflects that cancer cell is taking much more energy from the media and itself is very much fluctuating. Okay. Overall energy is increased. So, I mean this bioimpedance data can be analyzed further to get insight, some more insight phenomena. The other important thing that we have done using bioimpedance study is the cytotoxic response of the cell. So, here also we did the cell culture, okay, and do the bioimpedance, real time bioimpedance measurement. While doing that, at in between some time during the cell growth process, we add some drug and see what is the signature of my bioimpedance data. So, here it is the MCF7 and this is the case for this one MDA 231. We started the bioimpedance, a cellular growth started here, okay. And so it is going, so growth process is taking place. Now at this point means about say about after 30 hours, after 30 hours we start give a drug paclitaxel that with a different doses, dose 10 nanomolar, 15 nanomolar up to 30 nanomolar we have given at the different petri-dish, okay. And then also continues to measure the bioimpedance. What we observe that the black one, it is the control one, control means without any drug, and that the cell growth process is taking place further and further, it is going up and up. But for the red color with a dose 10 nanomolar with this drug, some of the cells have died, and so its impedance value had diminished. For the blue one, for the green and the pink one, where the maximum amount of dose is there, drug is there, so almost all the cells was died, and therefore the impedance value decreases further and further, okay. So, this is the result that we have achieved. So, we have seen that for this drug it affects the MCF7, okay, and that signature we can get it from the impedance study. Because today for the cancer treatment, all the pharmaceuticals companies are doing research for the chemotherapy, the different drugs are being they are manufacturing, but what is, how much is the utility, how much is the effect for those cancer line, they have to observe it. For that, and for that what they are doing? They are doing the cell culture, then giving the drug, then they are keeping it in the cell culture over the time, then they will go for the labeling based bioassay, and it takes longer duration, lot of chemicals are required also, okay. For the labeling study you require, it is not a cost effective, you require lot of valuable chemicals. What we can do, same thing, you start the cell culture, and after as the growth takes place here, this is the it is started and growth process is taking place up to 60 hours the growth process is there, okay. Now here at the 30 hours we have given the drug certain amount, and then we the cell culture, a cell growth process, we observe the impedance of the cell growth process. For the black one, the control one, it is going on up to 60, it is going on, but other cases it started diminishing, and for the highest value of the drug it has very minimum, it goes to the minimum impedance value. That means minimum impedance value means almost all the cells has died. So, what is the dose FA, how much is the dose we can give? From this bioimpedance study you can get it, as well as it will reduce your total timing, measurement time, okay. The other one also it gives the same results, okay. But here what we observe that as I told, MDA is much stronger than the MCF. So, here the same effect we have seen, but here the amount of the dose is much higher than this. Here it is only 10 to 30 nanomolar is sufficient to kill all the cells, but here it started with 100 nanomolar to 500 nanomolar, the same drug, because it is much more I mean aggressive compared to this one. That is why total amount of dose requirement is also higher. So, that has been observed using this bioimpedance study, and here also we did the wavelet based transformation techniques to get the different features of the cells, and that way. So, one way is that bioimpedance can be useful for your to study the different cell culture things, to study the what is the efficiency of the drug, how much amount we can do, and for that it is only the experimental based we have did, and further it can be extended for on the quantitative evaluation of the whole study also. Okay. So, with this I will stop conclude my session. Okay. So, any questions you have, please.
Sir, in this empathymetric monitoring you did, sir, did you progressively added the drug, or sir, it's like you added this 10 nanomolar and then waited for
Waited. We added here, at this point we given the drug of this amount, and then we observed the what is the impedance
till
value.
death.
Till death.
Okay.
Yeah.
And you repeated this 5 times for 5 different doses.
Yeah, we not, this is not the result for a single experiment. This is the result of a 3 to 4 different yes samples, you can say. Okay.
Sir, is it not possible to measure, like in normal cell line which we did, we introduced the drugs at different concentration in a 96 plate well at the same time. So is it not possible to introduce the drugs at the same time, because in
Here also we did it at the same time. Here if you observe this one, here instead of 96 I have say 6 numbers of well, and in each well you are culturing. Okay. Now you add 10 nanomolar here, 20 nanomolar here, 30 nanomolar here, like that. It is just like this, and also in the initial slide I have shown you one photograph where you have a 96 wells also, and nowadays it is available, commercially available also. We have made it in our lab with a small facilities, but commercially it is available also. To keep the all the experimental conditions same, you have to go for that.
Sir, in the starting you told that we use a DC voltage for this process. Whether AC can be used?
Yeah, whole thing is AC. Because if you let me go to this slide here, time average, where it is, oh this is all time average. Here it is, I have started with a 10 to the power 2 hertz, almost DC, but you can go extend up to 0 hertz, which will be DC only. Then you increase the frequency and go up to 1 megahertz or beyond that also. In their bioimpedance, it is not DC will not give you any signal, characteristic signal, because it is not penetrating the cell. Until and unless my probe penetrate the cell, I cannot get the signature of the cell. Okay. So, thank you.
Sir, in that fabrication, that measurement setup you have shown, so that one syringe pump is connected and it is fusing these cell lines through the device, right? Yeah, this one. Yeah. Is it possible to, repercolate the entire thing? So that kind of
Yeah, it is some cases it is required, because media whatever it is, in this case it is just wasting.
Yeah. Okay.
but in some other cases you can take these things media and
again it can
again,
be
yeah,
percolated.
yes, yes,
So it will
yes,
give more sensitivity. Right?
more sensitivity, yeah. But here I want to capture the cells.
Okay, and from this petri dish eluted thing, you are measuring the impedance. Okay.
Sir, no question. Sir, is there any difference in this cytoplasmic streaming in the normal cell and the cancer cell, and would that affect this impedance results? Is there any difference of the cytoplasmic?
Surely the cytoplasmic resistance will also changes as the disease progresses. Your whole thing, the cell, if we consider the cell membrane, and in the cell membrane we have shown that there is a capacitance, equivalent capacitance changes for the normal and cancer. Cancer cell, the capacitance is just a double to the normal. Because we have also confirmed that the ACM, from the ACM figure we have confirmed that you have a very much high roughness. High roughness means higher surface area, and that leads to the higher capacitance value. Now inside that cell you have the cytoplasm, and cytoplasm resistivity will also differs. Probably I have not put any slides on the what is the resistance value, but it will also changes. This is the capacitance change. I have shown for the HeLa cell it is 100, and for the Hacker cell this is the normal one, it is about 50, and the higher capacitance value is due to the higher surface area due to the roughness of the cell membrane. Okay. Now talking about the resistivity slides I have not shown, but they are also, you can from this data, yeah, this is the cell membrane you can calculate. This is the equation for the the cytoplasmic resistance value, how it differs it can calculate it. Where some of the studies we have also made, and we have observed that there is a change from normal to malignancy, there is a change. But of course these are in the research level. Can I take it and tomorrow can I, any industry under? No, we require further studies to establish this result. Okay. Much more robust measurement, much more validation it is required, and we have used only the cell line. And what about for the, if I take it from the animal, from the human, what is the result? Those studies are required. Then only yesterday I told, once I made a device in the lab, how many days it will take to commercialize? Tens of years, 10 years, 15 years, 5 years. As you have so many things. For biological or the biomedical, it will take much longer time, because it has to pass through that all the different stages of the clinical trials, human trials, then only it will come. Okay.
Sir, one more question. In the impedance based cell biosensors which you constructed, you used gold plated electrodes. Can these electrodes be used again after the process?
Generally not, because it is a corrosive, media is very corrosive, and so after 5 days you do not left anything more. We generally did not use it.
But is there a possibility to find an alternate for this kind of biosensor, because it is not cost effective, right? It can't be commercialized.
No, 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 it is copper. Why you think it is not cost effective? Because of the gold. But for your information, whatever media you are using, that is much more costlier than the gold, whatever, how much consumption of gold it is. It's a very thin layer, very small amount. Compared to that, your media, the cost of the media is very, very high, very high. Okay.
Sir, sir, for this cell, we need to consider the micro channels, because in the cell there are lots of channels. So, like sodium ion channels, sodium potassium ion channels. In this case, so here we are considering the current means only the electron beam. That means no the current due to the any particular flow of the ions, like sodium ions or the potassium.
No, that we have not considered. The way, because here we are considering the whole experiment we have done considering a cluster of cells. There are two aspects. Today people are doing research. One, it is using a cluster of cells. Another group they are doing in the globally using a single cell. Get the characteristics of a single cell. Okay. There also you put the between the two electrodes, put a single cell and see what is the impedance. Okay. Now, I believe that a single cell will not give you the proper picture, because our body, our human body or any living system cells having in a cluster formation, and their characteristics is evaluated in a when they are in a cluster formation only. That is why I did experience with the cell culture and all, and there we have not considered this these ion channels. We have not considered, because it is a cluster. The contribution from the single ion channels will be much less effective. Okay. If you consider a single cell, there you have to consider that ion channels also. Okay. But here you have a 10 to the power 6 number of cells, and the contribution from those channels will be negligible. We have not considered. Okay.
Because those microchannels create some kind of hurry up kind of thing.
Yeah, yeah. But I have not considered that thing, but it will be very less. But for the single cell, if you consider a single cell, suppose I want to measure IV characteristics of a single cell, that case you have to consider this thing. It will be much more effective. Say for electrophoresis, electrophoresis means between the between a cell you will put a two electrodes, and I will apply a voltage, so that voltage level should be very high, so that a pores will be created in the cell membrane. That is the by electrical means you are creating a pores in the cell membrane. Then I can add the drug, and that will go inside the cell through those pores. That is the electrophoresis. In that case, single cell electrophoresis is much more effective compared to cluster of cell. But here in the bioimpedance study, to get the characteristic features, characteristic properties of the cells, I will consider the cluster instead of having only single cell. Okay. So, thank you for your attention. Now, from tomorrow again onwards, Professor Science-Sina will take the class starting from 10 o'clock. And also I hope that all of you have got that my mail for the lab visit. So, Monday, Tuesday, Wednesday and Thursday, these 4 days we have made the 4 different groups, and we have a our specialties are scattered in different departments. In my School of Medical Science, probably you know or where is the location, or may not know some of you. It is a big campus. There I have some facility, moderate facility for the BioMEMS activity, and students they are working there. So, one lab is that one. Then Professor Soumen Chakraborty's microfluidics lab is located in the mechanical engineering department. Also in our I have another some facilities, microfabrication facilities in the Advanced Technology Development Center, and another facility, that is the electron beam facility and Raman spectroscopy, that is available in the CRF, Central Research Facility. So, 4 different locations we have the scattered facility. So, we have made the 4 groups. Probably shall I have to read the group names or not required? Not required. So, now for the group 1,